Καλό σας απόγευμα. Ελπίζω να με ακούτε. Ξεκινάμε μαζί με τη Βάσο στα Προεδρείο την καινούργια σειρά από τους...
Johnny, I have to interrupt you. We're English, yeah.
Okay, I think Yannis doesn't have a good, he didn't hear you, he's actually traveling. So let me start, do the introduction. So thank you all for joining today's seminar. Today is the first one from a new HIAAD seminar series with a different form than the one that we used to have. So the title of the seminars is Bridges, Γέφυρες, because our main goal is through these seminars to bring together basic and clinical researchers, people that interact with patients like health professionals and dissemination people, to present and bridge different aspects and perspectives in a specific scientific question, always related with the dementia and neurodegenerative diseases. Our goal also with these seminars is to promote new collaborations and synergies between the three main axes of HIAAD: the basic research, the clinical research and practice, and the patient care and dissemination, but also to include in these seminars also junior faculties or junior members that work in the labs of HIAAD members. First, let me present you briefly our speakers for today. So first, Dr. Savvas Christoforidis is a professor of biological chemistry and director of the Biomedical Research Institute of 4th at Ioannina. He got a degree in chemistry in Thessaloniki and in biological chemistry at Ioannina, and did his postdoctoral fellowship at EMBL in Heidelberg in Germany in the field of endocytosis and signaling. His work has been published in high-impact journals and has received over 4,000 citations. His main research interest is the role and mechanism of intracellular vesicular transport in vascular physiology and the implications in vascular diseases. Our second speaker is Dr. Eleni Aretouli. She is a licensed psychologist, associate professor of neuropsychology at the Department of Psychology and School of Social Sciences at the University of Ioannina in Greece. She has extensive research experience in Europe and United States. Dr. Aretouli's main research interests focus on the biological basis of psychological processes and the impact of various neurological and health-related conditions on cognitive abilities and psychological processes. The topic for today's scientific presentation and discussion is Alzheimer's disease prevention, clear your mind using your blood vessels. So thank you for both for the presentations today, and the floor is yours.
Thank you, Vaso, for this introduction. I just want to show you a picture of the city of Ioannina in the northwest part of Greece, which is the city where my lab and the lab of Eleni Aretouli located. So we are in this city and present to you the seminar today. It's our great honor, and we are very excited that we are the first to present in this new series of seminars, the seminars called Bridges, Research Bridges. So in this first seminar, the title of the presentation is Prevention of Alzheimer's Disease, Clear Your Mind Using Your Blood Vessels. Eleni Aretouli will first present clinical and epidemiological data on metabolic syndrome and cognitive decline. And then I will continue with the role of blood vessels in Alzheimer's disease. And I will try to link the lifestyle risk factors with the disease through blood vessels. So Eleni will start her presentation, and then I will continue on the basic research part of this joint seminar. Eleni, please.
Okay, thank you. I think, Saba, you needed to stop sharing your presentation so I can have mine. Okay, thanks.
Right.
Second. I hope you can see my presentation. Okay, so first of all, thank you, Dr. Macharaki and Dr. Christoforidis for the very nice introduction. I'm very glad to present this joint seminar with Savvas, the first from a series of seminars we named Bridges. And I hope by the end of this seminar, you will all have an idea why we named it this way. So the purpose of the specific joint presentation is to highlight the detrimental contribution of vascular pathologies, among other pathophysiological pathways, to cognitive decline and dementia. And I'm going to start my presentation by showing data from a recent systematic review we conducted on the association of metabolic syndrome with cognitive decline, and then a cross-sectional analysis again on the same topic. Then my colleague Savvas will continue by illustrating a pathophysiological mechanism that may potentially mediate this relationship and explain the clinical findings. So our rationale in joining efforts from clinical and basic research is to try to achieve an understanding of cognitive disorders based on the recognition of the underlying biological mechanisms. We consider this critical for the development of new methods for the prevention of cognitive decline and Alzheimer's disease. So I think there is no doubt that the mechanisms underlying late-onset Alzheimer's disease are multifactorial, and unfortunately, they are poorly understood. Among other factors, there is evidence that vascular dysregulation is one of the first of the early events on a cascade of events associated with the disease's progression. And metabolic syndrome is a risk factor for vascular and cardiovascular diseases, and also its individual components, which I'm going to mention in the next slides, such as hypertension, diabetes, hyperlipidemia, all share common features of compromised endothelial cell function. However, there is still no consensus on the link between metabolic syndrome and cognitive decline, so this association is not clear. So what is the metabolic syndrome, or as we call it, METS? So it's a clustering of commonly occurring disorders, and in particular obesity, dyslipidemia, hypertension, and insulin resistance. But we believe that METS is more than the sum of its individual components. And in fact, it is the synergetic effects of its components that is of interest, as it may exacerbate or influence in a particular way the effect of the individual factors, leading to more severe potentially consequences for cognition that would either pathology alone. And there are two additional reasons why one should investigate the metabolic syndrome. The first one is its high prevalence. So 1 in every 4 or 5 adults fulfill criteria for metabolic syndrome, according to the International Diabetes Federation. And also metabolic syndrome is strongly associated with lifestyles such as unhealthy diet, physical inactivity, which are modifiable risk factors. And modifiable risk factors are very important for the prevention of Alzheimer's disease and dementia. And here you can see that the criteria for METS are not exactly the same, depending on the health organization that proposed them. So these are the 3 most widely used criteria from the World Health Organization, the National Cholesterol Education Program's Adult Treatment Panel, and the International Diabetes Federation. And despite their similarities with regards to the basic aspects of obesity, dyslipidemia, hypertension, and insulin resistance, the different criteria often end up describing distinct clinical profiles. So there is a variability. And starting by the individual components that synthesize the metabolic syndrome, what we know with some certainty as far is that obesity and hypertension in midlife and diabetes, especially in later life, are significant individual risk factors for the development of future dementia and Alzheimer's disease. So the individual components of meds have already documented negative cognitive consequences. And according to the same report, the 2020 report by Livingstone, if we could minimize diabetes, treat hypertension, and reduce midlife obesity, then we could potentially reduce neuropathological damage, amyloid or tau-mediated, vascular or inflammatory, and thus prevent dementia. We undertook this, as I mentioned, although the meds components are already included in prevention guidelines for dementia, it's not clear whether meds itself, the metabolic syndrome itself, increases the risk of cognitive decline. So we investigated the association between meds and incident decline in overall cognition and on distinct cognitive domains. And initially, we aimed to perform a meta-analysis, but due to the very high heterogeneity in the methodologies applied in the distinct studies, that was not, unfortunately, possible. So we searched databases, PubMed and Scopus databases from inception to December 22, and you can see the search terms here. And basically, we included studies, human studies in adults with no serious psychiatric or neurologic diseases. The study had to refer to meds as a combined factor, not its individual components, and the outcome of interest was always cognition, but assessed with standardized neuropsychological procedures. Also, we selected only studies that utilized a prospective design. And here you can see the study selection procedure. I will not explain further. I think what is interesting is that we ended up including 30 studies. And those 30 studies included in total 62,471 participants. And you can see the heterogeneity of the studies as the samples ranged from 43 participants to 7,000 participants. So age at baseline cognitive assessment was over 50 years old, and most studies were conducted in Europe, followed by the US, Asia, Brazil, and one was multinational. And the follow-up period ranged from 1 year to 60 years. So the cognitive domains assessed in relation to meds was global cognition, which was assessed in 22 out of the 30 studies, memory in 90 studies, executive cognition in 60 studies, attention in 12 studies, and visual constructional abilities and language in 3 studies. And basically, our first finding is that over half of the studies concluded that meds may lead to faster rates of cognitive decline in at least one cognitive domain. But I should say that the results were rather mixed, though. And so here you can see an albatross plot summarizing in a narrative way the results we had. And on the x-axis, you can see the p-value of the association between meds and cognition. And on the left is a negative association, and on the right is a positive association. And on the y-axis, you can see the numbers of participants included in the distinct studies. And so what we observe is basically that there are a few studies like those ones that supported the negative association of meds with cognition. And I should say at this point that cognition was measured as a categorical outcome in this analysis. And also, most of the studies had null findings, so non-significant findings. And then a few studies, we're going to see that in the next slide as well, they reported positive associations. Here is a similar graph that summarizes the association of meds with cognition, but as a continuous variable. And we see some different results in a way. So we see more negative associations. The two positive associations over here remain. And the interesting thing is that those two positive associations basically were from studies that included very old participants over 75 and 80 years of age. So there may be an effect of age on the association of meds with cognition, which is not clear yet. And also, I'm sorry, I didn't show these results, but when we examined the potential interaction of age, sex, and a genetic risk factor for Alzheimer's disease, we have calculated, then basically we found no effect. So the genetic risk factor for Alzheimer's disease didn't change those associations at all. And the conclusion of this review is that the results are rather mixed. So there is some evidence that the presence of meds accelerates cognitions, but there's also some very limited, though, evidence that the presence of meds decelerates cognitive decline. And when we look in specific cognitive domains, not overall cognition, the majority of the literature shows non-significant results. So clearly there's a need for more studies with more standard methodological approaches. And probably not as to reduce heterogeneity in the methodologies applied. And next, we also conducted a cross-sectional analysis using our own data from the Greek cohort of the Epirus Health Study. This is actually a work under review. Our aim again was to explore the relationship between metabolic syndrome, its individual components with cognitive function, and to examine a possible interaction according to sex, age, and genetic risk factors for Alzheimer's disease. And you can see here a slide for the Epirus Health Study. Basically, the Epirus Health Study is a prospective cohort study designed to investigate the etiology of complex multifactorial chronic diseases. And this is, in a nutshell, the strategy of data collection and aims of the Epirus Health Studies, which basically combines information from questionnaires, neuropsychological tests, and molecular phenotyping. And this is the study sample included in the analysis. Basically, there were 2,077 participants included in the analysis. Their mean age was 46.6 years. Women and individuals of higher educations were overrepresented in the sample. And the 17% of the participants fulfilled criteria for metabolic syndrome. Compared to those without metabolic syndrome, individuals with metabolic syndromes were older, less often women, more often of lower education, and less physically active. And here you can see the cognitive performances, the neuropsychological performances of the participants overall and by presence of metabolic syndrome. And basically what we observed is that participants with metabolic syndrome had lower scores on measures that assess attention and processing speed, like the trail making test, verbal fluency, and also on memory measures. And similarly, when participants were assessed on computer-based tests, almost a year after their baseline assessment, we observed similar results. So participants with metabolic syndrome had lower performances, again on tests that assess attention, but also on a memory task, that an emotional word recognition test. Metabolic syndrome, in conclusion, metabolic syndrome was associated with poorer cognition in this middle-aged Greek cohort. And we believe that cognitive changes may occur early during the METS process. Given their lower level of cognitive functions, we hypothesize that those with the metabolic syndrome are at greater risk for dementia. And of interest is also that the metabolic syndrome may be a risk factor for decline in the absence of known factors, such as the APOE4 allele. And to summarize both studies, although the results are mixed, there is evidence of an association of metabolic syndrome with poorer cognition and cognitive decline. Though limited, there is some evidence. And the questions that remain are what may be the pathogenic pathways of cognitive dysfunction and dementia in metabolic syndrome, and also whether different pathogenetic pathways may explain the discrepant findings. And I would like to especially thank Dr. Genedis, who leads the Epirus Health Study, and his research team for the collaboration on this project, and also Mirtok Zonida, who is a PhD candidate who works with us on this subject. And I would like to thank you all also for your attention.
Eleni, please stop sharing.
Okay. Yes, sorry. Okay.
Okay. I hope you see my screen now. We have heard from Eleni already that there is a strong link between lifestyle risk factors like obesity and metabolic risk factors on dementia. There is a strong impact on dementia from these factors, but we already know that these same lifestyle risk factors have a strong impact on vascular properties, vascular function, and cause vascular diseases. So a very interesting question that arises in the field is whether the impact of lifestyle risk factors and other factors on dementia and Alzheimer's comes through a primary effect on the vasculature. So what we are interested in addressing is the role of the vasculature in Alzheimer's disease, and more specifically, the role of this particular cell type, endothelial cells. And to address the role of blood vessels in Alzheimer's disease, we needed to assemble a network of experts in different fields, as you will see in this presentation. And this became possible thanks to the HIAAD network, which brought together people from different fields. So we have participation from 2 institutes of Forth, from the Johns Hopkins University, from the Ioannina University, from the Institute of Demokritos, and from the University of Ioannina to address this topic. Our group, my group, is located in BRI Forth here at Ioannina, and we became interested in the Alzheimer's disease only very recently. Our main interest is on the properties of the vasculature and the role they play in inflammation and thrombosis, and more particular, the role of these cells, endothelial cells, lining up the inner layer of the blood vessels. And these cells are very well known to play an important role in inflammation and thrombosis, since we know very well now that when these cells are stressed by different stimuli or when they are damaged, there is extravasation of leukocytes towards the tissue, which causes the inflammatory phenotype that we see on the left, or the thrombotic phenotype that we see on the right. By green color, we see the platelets here aggregating and forming the blood clot, almost completely blocking the blood vessels. So these are the primary pathophysiological functions of endothelial cells and blood vessels. In addition, they play an important role in cancer as they supply the tumor with oxygen and nutrients to allow it to grow from small size to bigger size, which altogether the cardiovascular and the cancer diseases sum up to almost 70% of deaths in the human society. So in addition to all these roles of the vasculature, we have a very important role of the vasculature also for the brain. And I summarize the importance of the vasculature for this particular tissue in this slide. Brain consists of 2% of the body mass, yet it consumes 10 times higher oxygen levels and glucose supply in comparison to its size and in comparison to other tissues of the body. And to achieve this consumption, it requires 500 kilometers of blood vessel network with a total surface area of 20 square meters, which is actually the blood-brain barrier that allows exchange of important elements for the tissue. Given this dependence of the tissue in oxygen and nutrients, it becomes very vulnerable to damages when there is a shortage of blood supply. Its function is compromised, and we have appearance of phenomena like stroke and ischemia. There are some very interesting recent studies of single-cell RNA-seq data showing that the vasculature plays a very important role for the function of the brain in aging and in Alzheimer's disease. As we can see here, different functions of the cells and different cell types of the brain in the top. We can appreciate that endothelial cells, shown here in this red rectangle, we can see that most pathways of endothelial cells are compromised or are changed during aging. And if we compare the changes in endothelial cells and in other cell types like neurons and astrocytes, we will see that endothelial cells are affected at least more than those cells that have been the focus of brain diseases until now. So endothelial cells is a cell type that has been neglected despite its importance actually for the function of the brain. And if we analyze a bit further these consequences of aging in endothelial cells, we will see that the pathways that are affected include also metabolic pathways, regulation of lipid biosynthesis, hormone metabolism, citric acid cycle, and oxidative phosphorylation. All these are compromised while other pathways are increased. So endothelial cells is a target of aging. Besides, in an independent study, very recent study, where they have conducted the Human Brain Atlas in Alzheimer's disease, one can see the spectacular effect that endothelial cells have in terms of altered expression of genes associated already in Alzheimer's disease in genome-wide associated studies, followed by microglia, macrophages, neurons, and other cell types. So in these slides, I hope I have convinced you about the importance of these cells in brain function, but what remains to be answered is how do blood vessels get involved in Alzheimer's disease? What is the mechanism behind it? The hallmark of the disease of Alzheimer's disease is the accumulation of alpha-beta amyloids and tau, which destroy neuronal cells and causes the shrinkage of and the damage of neuronal cells and damage of brain areas. And there is already a correlation between the function of the vasculature and the removal of the alpha-beta amyloid, as I will show you in the following slides. So what we know already is that in a healthy brain, and if we zoom in a healthy blood vessel of the brain, we will notice that alpha-beta amyloid produced by neuronal cells is transported through these brownish cells that we see here, which are the endothelial cells lining up the internal part of the blood vessel. So the alpha-beta peptide is transported through the endothelial cells towards the circulation to be then transported towards the liver for degradation. And this is the function of a healthy brain, while in Alzheimer's disease, this transport of alpha-beta through the endothelial cells is compromised, causing an accumulation of alpha-beta in the area where neuronal cells are located, causing damage of neuronal cells. And to understand better the mechanism of how alpha-beta is transported through endothelial cells to the circulation, we need to make a bigger zoom of these cell types, of the endothelial cell types, and see how they achieve this function to transport alpha-beta to the circulation. So making a zoom of these cells, of these small cells here, we will notice that they are polarized cells having a basolateral side close to the neuronal cells and an apical side where the circulation is placed. So what endothelial cells do is they endocytose alpha-beta peptide from the basolateral side into endocytic vesicles, which then secrete their cargo, being alpha-beta, towards the circulation for degradation into the liver. And it has been very interesting that it has been found that in Alzheimer's disease, this pathway of endocytosis is compromised. In fact, adaptor molecules, which are very important for the generation of these vesicles, are reduced, their levels are reduced, so endocytosis does not take place and this function is compromised. However, this pathway of endocytosis, the clathrin-mediated pathway of endocytosis, is only one of many in endothelial cells. And in fact, in endothelial cells, there are not only many different types, but most importantly, in the presence of growth factors, one of which, the most important one, is VEGF. In the presence of VEGF, the main pathway of endocytosis is macropinocytosis, as I will show you in the following slides, coming from work that we have done in the lab recently. So in the absence of VEGF, the receptor of VEGF internalizes into small vesicles that we see here by confocal microscopy. But when we knock down clathrin, we block this clathrin-mediated endocytosis pathway, we block the internalization of VEGF receptor 2. But we achieve this only when the growth factor is absent. In the presence of the growth factor, in the presence of VEGF, despite knocking down clathrin, the receptor internalizes. So if you look at this picture here, down here, and this one, there is a big difference. In the presence of VEGF, it is impossible to block internalization of the receptor by blocking down clathrin-mediated endocytosis, suggesting that when the growth factor is present, the receptor internalizes through another pathway independent of clathrin. And we have identified this pathway as macropinocytosis by video microscopy and another experiments, showing that in the presence of VEGF, we have ruffling of the plasma membrane, seen here in green, by actin GFP staining. And this is where the vesicles of VEGF receptor are internalizing. Here in the zoom area, we see better the vesicle in red being formed. I would stop the video and play. here like that. So you see in green in the zoom, in the top right side, you see in green the ruffling of the membrane and the formation of a green vesicle having actin around it and then becoming red as actin disappears and the receptor internalizes alone here. Stop the video so that we can appreciate it. The size of this vesicle suggests that it is a macropinosome, and there have been many experiments that we have done to further confirm this by electron microscopy experiments as well, showing the large size of these vesicles above 500 nanometers, which is the size of macropinosomes, which we also see here, their large size, which is above 600 nanometers, close to 2 micrometers a few times. All of these data suggesting that in the presence of the growth factor, the main pathway in endothelial cells is the macropinocytosis, which is also important for signaling, since when we block this pathway with a known inhibitor called APA, signaling of VEGF towards ERK and TACT is almost completely compromised, as we can see here, the quantifications. Also, angiogenesis in mice by VEGF is inhibited when we use the inhibitor. And all these data suggest that the clathrin pathway is active only in the absence of the growth factor, while in the presence of VEGF, the main route is macropinocytosis, which now in our recent interest on alpha-beta clearance and Alzheimer's disease makes the story interesting, as it appears that the clathrin pathway is not the main one in these cells. In the presence of VEGF is macropinocytosis, so it would be interesting to address the role of all these pathways in taking up alpha-beta peptide into the circulation for degradation. As our expertise is only on endothelial cells, to address this question, we needed also the contribution of people from the neurobiology field as well as from other fields. And all together here, you see the network of people contributing to this work using in vitro model systems, trafficking assays of alpha-beta peptide, animal models, transgenic animal models, and 3D collagen scaffolds, as well as bioinformatics analysis. In red, you see the different institutions contributing to this work. The Johns Hopkins University, Forth, the Institute of Democritus and Ionian University, and the people who are also contributing to this work are also mentioned in this slide. I will mention it also in my last slide. Already we have submitted 2 proposals for funding so that we can proceed this work. We have submitted to ELIDEC, a Greek funding agency, and Marie Curie, a European one. Evi Vassiliki has contributed a lot from our lab to the writing of these proposals. And I will say a few words about this in vitro model system, as it gives us the possibility to do experiments mimicking what is happening in human brain. It's impossible to do experiments in humans in these types of experiments. So we need in vitro models where we place endothelial cells in membrane inserts. And at the bottom of these inserts, we can place neuronal cells producing alpha, beta, and tau, so that we can measure then the transfer of the peptides towards the apical side of these cells, mimicking what is happening in a physiological blood vessel in the brain. We have already established this system in the past, in previous studies, and these cells indeed achieve polarity, as you see 2 different markers, an apical marker and a basolateral marker, the apical in red, the basolateral in blue. They are completely separated in these colors. Here you see a cross-section where the apical marker is at the top and the basolateral at the bottom, suggesting that the system works well. And we need now to use this system to address the pathways that are responsible for the uptake of alpha-beta peptide. The right system to use to approach this question in Alzheimer's disease is to use, of course, cells coming from patients, and thanks to the iPS cell technology that has become available in the recent years, this is possible, taking samples from the peripheral blood of patients. And here we have to acknowledge the great contribution of Vaso Macharaki from the Johns Hopkins University and of Kostas Liketsos, who are doing the clinical studies and the production of iPS cells from peripheral blood cells. These cells then will be differentiated towards endothelial cells and neurons and will be used in this in vitro model system to study the endocytic pathways that are responsible for the uptake of alpha beta. And using this system, we can also test not only the endocytic pathways, but also the effect of lifestyle risk factors like LDL cholesterol, mimicking the lipid stress conditions, high glucose and insulin, conditions that are found in diabetes and hyperinsulinemia, and also to test the role of aging, comparing aged endothelium versus young endothelium of different passages of endothelial cells. And also to check whether the Alzheimer's disease plasma from patients is whether the plasma from Alzheimer's disease patients has different properties in comparison to healthy plasma, whether the growth factor levels are different, and what is the impact of those factors in the uptake of alpha beta in our system. And there we will collaborate with Eleni Aretouli and the Iberos Health Study, where they have already assembled a lot of these samples from patients well characterized. And using this in vitro system, we have already established the protocol for uptake of alpha beta. Here we see endothelial cells, human primary endothelial cells, not brain yet. They are from umbilical cords, but we needed that in order to establish the protocol. The cells are incubated at different time points with alpha beta peptide in green, 30 minutes, 2 hours, and 4 hours. And we see that the cells become more and more greeny as the time passes, which is due to the accumulation of alpha beta peptide in large vesicles inside these cells. These vesicles now need to be characterized, their identity, so that we can find the exact pathway of internalization. This internalized alpha beta is in large multimers, as you see here in this Western blot, above 200 kilodaltons. It doesn't enter the gel, while the monomer of alpha beta that we add to the cells is here. So when it is internalized into the cells, it forms large oligomers. So it mimics more or less what is happening in the physiological situation, this system. And in summary, concluding here, I conclude here by saying that in this in vitro model system, combining different expertise of iPS cells, bioinformatics, material sciences, model organisms, and clinical data, we hope to be able to address the exact pathway of internalization of alpha, beta, and tau under physiological conditions and in diseases. What happens, what is the defect in patients, and test the effect of risk factors. And here I would like to acknowledge the contribution of the people in this presentation, Evi Vassili, especially Evi Vassili, who has already established the protocol for the uptake and the detection of alpha beta in endothelial cells and has contributed to this proposal that I told you in the writing of these proposals, as well as different labs that have contributed to our work throughout years. But most importantly, I would like to thank our collaborators in this new project for us in HIAAD, especially Vaso Macharaki and Kostas Liketsos from Johns Hopkins University for the generation of iPS cells, which are going to be combined in this in vitro system that I told you. From IMBB, fourth, Ioannis Haralabopoulos and Nektarios Tavernarakis for the animal models that we are going to use to validate the findings from the in vitro experiments. Yannis Sotiropoulos for the expertise in tau, which is an important risk factor for the disease. Eleni Aretouli for the epidemiological and clinical data locally here from Epirus. and the Ionian University, especially Takis Vlamos, Themis Exarchos, and Aristidis Vrachatis, who were convinced initially from this idea and supported it so that it could start already some time ago. And thank you for your attention here.
Well, thank you so much, both Eleni and Savvas. There were excellent presentations. I would open the floor for questions. I can see already 2 questions at the chat. So Kostas Liketsos has a question. Kostas, we'll start with you. Do you want to do to make the question? I can, or
well, I can ask maybe a slightly different question. My question to Eleni as she was presenting is the extent to which METS is associated with sleep disorder. People who have METS have really poor sleep, and so the question is whether that was an effect modifier or confounder that was examined either in the it wasn't a meta-analysis, but in the literature spectrum or in your own research, because I think it's an important confounder.
Thank you for this question. So we haven't examined this factor in the systematic review, but we did examine. So we have data on sleep in the Icarus Health study, and in a separate analysis, we did examine the association of sleep with cognitive performance, and interestingly, there was no association. And also, when we included sleep as a variable in the association between METS and cognition, the relationship didn't change. So it seems that in our sample, I don't know in the extant literature, but in our sample, it didn't have any effect, sleep. But I think probably we should look into the maybe a summary of other studies to kind of have a better idea.
Thank you, Eleni, for the answer. I can see the next question from Nick Skarmeas. Nick, do you want to make your question?
Yeah. Well, yeah, it was just for Eleni. First of all, I want to thank Savvas actually, because not always do I understand, presentations from basic research. And I really enjoyed it because, I understood much more than usual. You explained everything so nicely. So the question was more to Eleni, whether when you said the genetic risk score, I put it in the chat, you meant ApoE or you estimated polygenic risk scores from GWAS from the study? Yeah.
So basically, the genetic risk score was included in our cross-sectional analysis, and it was polygenic. So based on the Janssen, I think, meta-analysis of 2019, there were, let me find this info. So there were 29 single SNPs included. And they were calculated as a genetic risk score. So it was not only ApoE.
Okay, thanks. Maybe I can add just here, so it was a polygenic risk score for clinical dementia, right?
Mm-hmm.
Right, so...
For Alzheimer's disease.
Okay, for clinical Alzheimer's type dementia then. So it may be, since you have this data, it may be worthwhile, exploring towards, constructing other polygenic risk scores that may tap into particular pathologies or biological pathways. So it's doable, for example, to calculate a polygenic risk score for genetic propensity for amyloidosis or for tau or for white matter hyperintensities or for inflammation. And it may be, the effect, the associations between the metabolic problems and cognition may differ as a function of these underlying pathologies. You never know, but since you have this data, it's really great. It's not available. It's not so easy to calculate, and it takes a lot of effort.
Okay. So thank you for this idea. I think we will go for it, and you can collaborate with us on this analysis.
Thank you. I can see Maria Basta has another question.
Hello, I would like to congratulate both presenters for their very clear and interesting presentations. I have a comment and I have a question because Dr. Liketsos opened the sleep discussion. I got really curious about that since I love sleep. So I wanted to share with you some findings we had in the Cretan aging cohort, where we unexpectedly found that those with metabolic factors had higher MMSE scores, which was unexpected. So that was in the first phase of our cohort, where metabolic factors like hyperlipidemia, etc., hypertension, etc., were defined based on a history or if the people were on medications for these diseases. So when we didn't expect this finding, of course, but when we found it, we were trying to understand it and explain it. And what we thought that could be an explanation was that maybe those were there were those that were diagnosed and treated, and their metabolic indices were lower because they were under treatment, and other people who had never been diagnosed or they were not taking medications for that. So I would like to ask you how you define. I mean, did they have lab testing? How did you define metabolic syndrome? And if you can make a comment on that. That's the first thing. And the other thing is, could you please tell me a little bit more about your sleep data? Like, are they subjective sleep based on questionnaire, sleep duration, quality of sleep? Because as Dr. Liketsos said, sleep is definitely associated with metabolic syndrome and especially sleep apnea. And Dr. Wondel has done a lot of work on that. So I would like to know what you have about sleep. Thank you.
Okay, I think this question goes to me as well. So basically, sleep was assessed based on self-reports, so questionnaires. So that might be one of the reasons we didn't get any association. I don't know, but it was just self-reports through questionnaires on sleep habits. Duration of sleep was included, but again, self-reporting. And then METS was diagnosed based on lab results. So I don't have the exact info, but I know that there were blood samples taken. So there were lab results. There were some anthropometric measurements like circumference, and so there were actual lab tests and assessments.
Kostas, Liketsos, do you have one more question? I think you have raised your hand.
I have, but I don't want to take away someone else's opportunity.
Okay, I will go to another question then first to Kostas Tsilidis, another Kostas. Kostas, are you in the call? Do you want to, I can read your question, or if you want, hear his again.
I'm in the call, but I'm staying silent since I'm not
Okay.
at all an expert in Alzheimer's disease or dementia in general. So I'm just the PI of the study that Eleni mentioned. So I'm just replying to the comment that we use the Pittsburgh Sleep Quality Index, which is probably the most known or the most used scale to assess sleep disorders. It's questionnaire based, but yeah, this is what we used for sleep. And regarding the other comment about treatment, which Maria made, which I think makes absolute sense in how they explain their findings. In the Ipirus study, most of the participants are middle-aged and without diseases. So most, not all. So I wouldn't expect this to be to make a lot of difference in our sample. Thanks.
Thank you, Kosta. So, Kosta Tliketsu, do you want to make a
brief comment? First of all, I want to compliment Yanni especially for pulling this together, and Eleni and Savvas. This is a really wonderful discussion and a terrific illustration of the power of the Bridges series. So I look forward to continuing this every month for a little while now. So thank you. My question goes back to precision medicine, and it goes to the issue of whether it is reasonable to expect that vascular pathology would be an influence on dementia in every individual or not. And whether we have ways of thinking how to differentiate individuals for whom vascular pathology is more or less influential on the dementia or Alzheimer's outcome. You pick your favorite outcome. So the example that we have that we're sort of pursuing right now depends on measurement. It's possible to measure brain perfusion on MRI, and it's also possible to assess how perfusion changes under a stress condition, which is inhalation of CO2. So ordinarily, when people breathe CO2, on an MRI, you will see an improvement in brain perfusion regionally and globally. But a team from Hopkins a few years back reported that in people with mild cognitive impairment, there's a subset of people who have a paradoxical response. So that you measure their baseline perfusion, you expose them to the stress of CO2, and this subgroup of people now has a reduction in perfusion, as if the brain not only isn't resilient to the stress, but goes the other way. So that's an example of starting to think of how would we define people as a subgroup who are vulnerable to the effects of vascular factors on brain. So Savva, I would encourage you to think, well, how do you do that in the lab, right? I mean, one of the reasons to do iPS cells is that you can look across many different people's iPS cells. But then you got to think of, do you pick those people for a polygenic risk factor score that means they are more vulnerable to vascular disease or Alzheimer's disease? So I think I'm getting the point across. I think this is very important because as a field, we've been doing general epi and not attributable epi. We need to start thinking of a risk factor specific influence on an individual as opposed to a global risk factor effect on a population.
Right. I think you said, Kostas, you said everything. Perhaps I don't need to add anything to that. Perhaps I could add only that what I have described here, the example that I described, the endocytic defect in this particular case of Alzheimer's disease, the one that has been described until now, the defect in clathrin-mediated endocytosis, was found in a particular patient from whom iPS cells were isolated, were generated and differentiated into endothelial cells and neurons, and they found that the components important for clathrin-mediated endocytosis were reduced, and alpha-beta peptide uptake was reduced. And they were able to correct this by increasing the levels of the defected proteins. So for a subset of patients, I would expect that indeed the compromise of the function of the vasculature would be the cause of the disease, but not for every single patient. That's why it is important to screen for different causes of the disease for each patient, so that one then can correct the defect of for every single patient individually. So I completely agree. I would expect not all patients, of course, to have the same defect in the uptake, but the example I showed you was actually very spectacular how important was the endocytic pathway. And in fact, those, I wouldn't expect perhaps those cells to, those blood vessels to have in early stages defects in, let's say, perfusion issues. Perhaps one would not be able to see perfusion issues in the early stages of the disease. This would come up perhaps later when alpha-beta amyloids accumulate, but one would be able to see earlier that the uptake of the peptide is compromised, and then perhaps could prevent, could act in a preventive way by activating endocytosis. And there are drugs that can do this. But all this is, until now, it is only in the basic research stage, of course.
Thank you, Savva. I can see one more question from at the chat. I don't know for whom. I guess it's for Eleni. It says, do you consider a relationship with lymphatic system?
It's for Savvas. Sorry,
It's
it's
for
me
Savvas.
because on the phone it's for Savvas. Sorry.
Oh, okay, for Savva, sorry. I mean, both of you, of course, you can reply, you can answer the question. Savvas?
I don't know what to say on that. One cannot exclude it, but it should be tested. One would need to set up in vitro systems to test this hypothesis, to assemble a more complete tissue, perhaps using the scaffolds, the tissue-engineered scaffolds, so that can accommodate more cell types that will include also the lymphatic system and address whether it is also involved into the amyloid uptake and removal and the dysfunction of endothelial cells. One would need to test this. It is likely, but I don't know. I haven't seen anything on that in the literature. Perhaps somebody else might have a better answer on this.
Thank you, Savvas. Are there any other questions or comments? Anything? It's already, of course, 11:07 here in the United States. I don't see any more questions. So I would like to thank everyone for participating in this first Bridge HIAAD seminar series. And I would like also to thank Eleni and Savvas for these great presentations. And I think we achieved our goal in the seminar, in the first seminar, how you can bridge and address a question, a specific question from the clinical point of view and from the basic research point of view. So thank you all for being here and participating.
Also, I don't know if you hear me.
No.
I'm on the train. Can you hear me?
Yeah.
Now, yes.
Just saying that in the next in the 1 month from now, we will have another seminar bridge between a basic biologist and a clinician that we will describe the connection between chronic lifetime stress and dementia. And this will cover both basically and clinical work, as well as a new initiative, a new clinical, let's say, attempt that is based on HIAAD interaction. And it is March 2, again, 5 o'clock Athens time, 10 o'clock Baltimore time. And you're welcome, please, when you receive the announcement, to please forward to all your students to overlap, because one of the main, let's say, aims of this bridges, the new seminar of HIAAD, is to bring more people and new members and young members in the HIAAD consortium and initiative. Thanks again, and sorry for not being able to be connected all the time. they changed my flight last minute. Thank you.
Thank you so much. So see you next month. To be continued. Bye. All.
Thank you.
Bye,
Thank you. Bye.
Bye, everybody.